This video is going to talk about the remaining types of vaccines that we are going to cover in this course. So next up are subunit vaccines. And with this type of vaccine, we are only subjecting the person to part of the pathogen. Okay, with both the live attenuated and inactivated killed vaccines, the entire pathogen was involved. Now what we're doing is essentially isolating that antigenic portion of the pathogen and including that in the vaccine. So we can make this type of vaccine by either chemically breaking down the pathogen and isolating the antigen, or we can use genetic engineering to only produce the antigen. What's really nice about these types of vaccines is that the risk of side effects is pretty low because you're not dealing with an entire pathogen, you're only dealing with the antigen. So, excuse me, with these subunit vaccines, there are a couple of different types. So with toxoid vaccines, these are mainly going to be used when the disease is based around the exotoxin. So we talked about exotoxins and how a lot of them are enzymes that just wreak havoc on the host. And normally it's the toxin that's really dangerous, not the actual bacteria itself, because the main weapon it uses is the exotoxin. So when the toxin is the concern, not the microbe itself, then vaccines that are made of toxoids are going to be used. So a toxoid is an inactive toxin. So normally we've modified the toxins by exposing them to heat or some sort of chemical agent that makes them harmless. And the challenging part of creating a toxoid vaccine is we need the overall structure, like the overall 3D shape of the the toxoid, to be similar enough to the active toxin that our immune system will recognize it. But we need the structure to be different enough that it's not harmful. Okay, so there's a lot that goes into producing this to maintain the overall 3D shape while changing it just enough that the toxoid can't harm the host. So it's a little kind of a puzzle there. So some of the toxoid vaccines that are used are diphtheria and tetanus, which are both part of the DTaP vaccines. Those are the main ones that we are going to be talking about. Another type of subunit vaccine is a recombinant vaccine, and the recombinant portion of that lets you know that it is genetically engineered. So we use various types of recombinant DNA technology and genetic engineering to produce the antigens that are found in viral capsids or other virulent factors that are part of the bacterial cell wall. And we make it so that bacteria that are not harmful are able to produce these specific types of antigens. So in order to do this, though, we have to be able to determine the genetic code for those antigens, right? We have to be able to identify what region of DNA is going to then produce the RNA and the protein that is the antigen, which can be tricky. It can cost a lot of time, energy, and money in order to do these. But once we know what the genetic code is, we can put that gene into a plasmid. Remember, that's a circular piece of DNA that bacteria can use. And so then those plasmids are given to either bacteria or yeast that will produce the antigen as they go through their normal, transcription and translation processes. So now we have ample amounts of antigen, okay, that can be injected as a vaccine into your body. And then, we'll have our immune response to those antigens. So these vaccines, kind of like the other ones, don't really pose the same risks as other vaccines because you're not going to actually get the disease from the vaccine. Now, you normally don't get the disease, but like the signs and the symptoms associated with the disease, you don't normally get any of those. There's no chance of anything reverting and becoming dangerous. So it's a nice, safe vaccine. Some ones you've probably heard of before are going to be the acellular pertussis vaccine, which goes after the organism that causes pertussis, aka whooping cough. And then the Gardasil vaccine, which targets HPV, is also a recombinant vaccine. All right, next up are going to be conjugated vaccines. And so this is a type of subunit vaccine that is going to help against pathogens that contain a capsule, because capsules are sugary, right? They're polysaccharides. And polysaccharides are just not very good antigens. They don't really trigger the immune response like proteins do. So to kind of get around that, we can essentially attach a protein to a capsule sugar. And because remember, proteins are very good antigens, it will stimulate more of an immune response. And in turn, our immune system will recognize both that protein, but also the capsid protein that it is attached to. So it's kind of a nice way that we can get around these pesky little capsules, sorry. And I just realized I skipped over one of my other subunit vaccines, so I apologize. Another type is a virus-like particle vaccine, but essentially the components in the vaccine resemble an intact virus, but there's no viral genetic material. So again, the body is trained to recognize that overall virion structure, but the actual dangerous part of the virus, right, the genetic material, is not actually present. So our immune system will recognize the structure of the virus when we do come into contact with it naturally, and we'll already have an immune response ready. But with the vaccine, there's actually no threat of a real infection because there's no genetic material. So it's nice and safe. Okay, so let's go ahead and wrap this up with a talk on nucleic acid vaccines. So I apologize, that DNA there in parentheses should have been deleted. I moved it to a bullet here. So please go ahead and make it so that this second bullet point just says nucleic acid vaccines. So this is where we are going to be including genetic material from a pathogen, or that mimics that of a pathogen, into the vaccine. So that way, once it's inserted into the host, transcription and translation will result in the antigen being expressed. So that way it'll trigger our immune response, and we'll get a nice full-blown immune response, which involves memory cells. So it'll be a nice, more comprehensive, long-term immunity. So DNA vaccines specifically are relatively new, but they are promising. Same goes for mRNA vaccines as well. With a DNA vaccine, what happens is the DNA that is specific for the pathogenic antigen is put into a plasmid. And then when a person gets injected with that vaccine, then that person's cells are going to do transcription and translation. And that antigen is going to be made and presented along with MHC1 molecules, which is going to activate our adaptive immune response. So that means that this type of vaccine is going to activate humoral and cellular immunity. But because we've only expressed the antigen, we don't have those risks that are associated with live attenuated vaccines. So that's a huge perk. Now, most of the DNA vaccines that exist are still in development, but it's very likely that they're going to be much more prominent in the future. Because again, there's just, it's eliminating that risk of, potentially getting sick by having a live weakened pathogen as part of the vaccine. You've probably heard of the mRNA vaccines because that's what the COVID vaccines are, which is why this is a really nice, safe method to developing immunity towards COVID, or at least, greatly reducing the risk of getting sick. And so it's very similar to the DNA vaccines, except we're skipping the DNA part. We're going straight to the messenger RNA. So that way our body is going to naturally produce the antigen, which will then trigger the immune response. And just like with DNA vaccines, we're going to get a nice full-blown response with those memory cells. So we get a nice long immune memory. There we go. That word was evading me. All right, y'all. That is going to be the end of this video. Please let me know if you have any questions.